Okay. Sound check, students. Let me know in the chat if I am audible and visible to everyone quickly. Hello students, welcome to Vedantu NEET English. This is Baswaraj sir, your biology master teacher. So quickly let me know in the chat if I am audible and visible. I am in a different studio, so it is very important to note, right? If I am audible and visible to all of you. Students, this is the second chapter from the line by line series. So all of us, we conquered the first part, that is going to be your cell the unit of life. Every single point to point NCERT we are done and dusted. And we also solved a lot of pyqs. So students, those of you who missed out the last part, right, please go watch it. Please go watch the cell the unit of life last part, part 3, which has a lot of pyqs. Now students, when I was reading your comments, I realized that you want to do numericals, or you want to do questions as soon as the class is over. You do not want to wait until the very end, right? All of you want that. So for that very reason, I have decided that every class onwards, now if today is the class, we'll be doing cell cycle and cell division part 1. In this class itself, we'll be solving pyqs. How does that sound, right? How does that sound, students? So today is the class, we'll be doing the cell cycle and cell division part 1, and we'll be solving pyqs in today's class only. So we do not have to wait until the very end. So that sounds amazing to me also. So with that being said, students, can we start today's amazing class that is going to be cell cycle and cell division? Now tell me honestly all of you, how many of you know this concept of mitosis and cell cycle 100%? Raise your hands quickly. All of you raise your hands who think they know the entire concept of, mitosis as well as the entire concept of cell cycle 101%. You cannot make any mistake. Tell me, raise your hands, quickly raise your hands, all of you. And those of you who are raising hands, quickly mark your attendance also. Quickly mark your attendance also. Now students, let me tell you, NCERT is the key, right? NCERT is the key. While you read NCERT, there is a concept called as DNA content. There is a concept called as DNA content, which that also I'll be teaching in today's class, right? So what is this 2 and 2C, 2 and 4C, 4 and 4C? All that DNA content I'll be teaching you today also. So you don't have to worry a bit. So let's start the class with the NCERT reading. So all of you quickly open your NCERT with you. Keep a pen next to you and keep a, okay, I missed my water bottle. Keep a water bottle also next to you. I don't know where I kept my water bottle. So keep a water bottle, NCERT, a pen. So you write down the points which I tell you. Okay. Quickly let me know in the chat if you're ready. Yes. So students, let me know if it's visible properly. Because if there's an increase in issue in brightness, I will try to adjust it. Okay. Now let's read the lines of NCERT, all of you, quickly. Cell cycle and cell division. Now let me tell you students, the entire chapter, especially the mitosis, it tells you how a cell divides. It tells you how a cell divides. That is 1 cell to 2 cell. 1 cell to 2 cell. Okay. Are you aware that all organisms, all of you only, even the largest, start their life from a single cell? Now students, what is the single cell are they referring to? Can anyone tell me what is the single cell are they referring to? Is it the zygote? Is the single cell they're referring to here the zygote? Yes or no, quickly in the chat. Zygote. If it is the zygote, tell me the zygote right here, is it diploid or haploid in the nature? Quickly. Yes, the zygote right here is diploid in nature. Yes, it is diploid in nature. Now, you may have wondered how a single cell then goes on to form such a large organism. For example, Rashmita here, Pavitra here, Jensly here, all of you started your life with a single zygote. All of you started your life with a single zygote. Later on, as you grow older, from one simple zygote, your life changed. You grew into a complete adult, right? So all of you high zygotes. Okay. Now, growth and reproduction are characters of cell. Yes, growth and reproduction are characters of a cell. Now students, this line reminds me of an organism. This line right here reminds me of an organism where growth and reproduction are same to same. Do you remember also? There is one organism where growth and reproduction are synonymous. Meaning is that as the organism is growing, it also reproduces. If the organism is reproducing, it is also growing also. Can anyone tell me which is that organism? Which is that organism where growth and reproduction are side by side? They go hand in hand. If they reproduce, they also grow. That is your exactly. Those are your unicellular organisms. Growth and reproduction are synonymous in the case of unicellular organisms, unicellular organisms. Simple example is going to be your bacteria. Simple example is going to be bacteria. All of you remember that, right? All of you remember that. Amazing. Now students, okay, indeed of all living organisms, all cells reproduce by dividing into two. That is, from one cell, students, from one cell, it divides into two daughter cells. One cell to two daughter cells. Okay, two daughter cells. These are the daughter cells. From one cell to two daughter cells. Okay. Next point, student. Next point. Very easy and understand. Okay. With each parental cell giving rise to two daughter cells each time they divide. So one parent cell dividing into two daughter cells. Now, the newly formed daughter cells can themselves grow and divide and give rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. So basically they are trying to tell you is that now this cell, student, this cell right here, this cell will eventually grow. It will grow in size, increase in the cytoplasmic content, and this cell will also divide into two daughter cells. Again, this cell will also divide into two daughter cells. This cell also will divide into two daughter cells, and that's how the journey of cell division starts. Journey of cell division starts. Amazing, amazing. All of you know that, right? All of you know that. Now, all this talk about cell cycle and cell division, it reminds me of a concept called as cell theory. Quickly tell me in the chat, who were the two scientists who initially gave the cell theory? Quickly in the chat. I'm checking in the chat. I'm constantly looking at the chat, students. Do not cheat. Okay. Who were the two first scientists who gave the cell theory? It was Scheiden and Swan. Scheiden and Swan gave the cell theory, and they told us that all the living organisms, plants and animals are made up of cells and product of the cells. They are made up of cells and product of the cells. But they could not answer one question. Can anyone remind me what is that one question they could not remind, and one more third party had to come? That is, Rada-Furchow had to come and tell that you are not able to figure out. You are not able to figure out that how did the cells come into existence? That's right. How did the first cell come into existence? It is like your chicken came first or egg came first. Almost similar but not same but different. Same but different. Right. Now this theory was given by Rada-Furchow. He told that all the cells exist from pre-existing cells. They come from pre-existing cells. Together they gave the cell theory. All of you remember. All of you remember, right? Nice, nice. Now, in other words, such a cycle of growth and division allows a single cell. Growth and division allows a single cell to form a structure consisting of million cells of all of you. All of you are consisting of millions of cells. Agree? Gently, it could be Aparna, it could be Ahmed, Madhumati, Mukundas. All of you are made up of millions and millions of cells. So students, tell me in the chat quickly if any doubt in the first paragraph. If there is no doubt, let's go to the next paragraph because it is a very easy chapter. Very easy chapter. Right? Now, next paragraph. Now students, now we'll come to cell cycle now. We'll start with the cell cycle, the entire cell cycle. Okay. The cell cycle is a very important process in all living organisms. Okay. NCERT is only telling you, so we'll mark it. Very important. NCERT is telling you, we'll mark it. Very important. Very important. Okay. During the division of a cell, DNA replication and cell growth also takes place. So students, if they ask you what happens during division of a cell, during division of cells, what happens? DNA replication takes place. What happens, students? What happens? During division of a cell, what happens? DNA replication takes place. DNA replication. And apart from the DNA replication, second point is what? Second point is going to be cell growth. Second point is going to be cell growth. So if someone asks you, students, if someone asks you what exactly happens during the division of a cell, you should be able to tell me, sir, DNA replication takes place as well as cell growth also takes place. Cell growth also takes place. Done? Done? Yes. Now, let's go to the next point. Okay. All these process, cell division DNA replication and cell growth. Hence, have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genome. So, all they are trying to tell you here is, first a cell divides. First a cell divides. In dividing, when a cell is dividing, there is DNA replication. Then the cell grows in size. Now, imagine you're trying to make biryani. Imagine you're trying to make biryani. Biryani is made in layers. Biryani is made in multiple layers. All of you know, right? We add some rice, then we add some chicken, then we add some rice. On the top, we add some onions, caramelized, crunchy onions. Can you, if you make it ulta pulta, will the biryani come out proper? No. Or you're trying to make some kind of dosa. Instead of adding water on top, if you add directly dosa batter, will dosa come properly? No. Everything has to be in a coordinated fashion. Everything has to be in a proper fashion. Okay. If there is something ulta pulta, your biryani is also not happening, cell division also is not happening. Okay, now, the sequence of events by which a cell duplicates its genome, genome is basically the DNA content, synthesizes the other constituents of the cell and eventually divides into two daughter cells is called as a cell cycle. So, basically what is cell cycle, students? Cell cycle is a cycle in which there is division of a cell, growth of a cell, division of a cell where we replicate the DNA and the cell is also growing in size. See, that is the sequence. It is a sequence of events by which a cell duplicates its genome. That is DNA replication. Synthesis the other constituents of the cell. That is basically it could be Golgi complex or it could be endoplasmic reticulum, nucleolus, all the other constituents, right? And eventually divides it into two dotted cells is called as cell cycle. Understood? Understood. That is called a cell cycle. Okay. Although cell growth in terms of cytoplasmic increases in a continuous process. Oh, hidden line of NCERT. Hidden line of NCERT. Line is very simple. It is telling you that although the cell growth in terms of cytoplasmic increase is a continuous process. I'll ask you a question now. Cytoplasmic increases, is it continuous or discontinuous? Go. Simple question. The cytoplasmic increase in the cell, is it a continuous process or is it a discontinuous process? Ask. Could tell me the answer. The cytoplasmic increase, is it a continuous process or a discontinuous process? Very simple line, you can always miss it out. That is the importance of line by line. See here. So, it is a continuous process. Now, DNA synthesis occurs only during a specific stage in a cycle, contradicting to the cytoplasmic increase. Cytoplasmic increase happens continuously, but your DNA replication takes place in a very specific point in the cell cycle, and that is called as the S phase. That is called as your S phase in the cycle. It's called as the S phase. Also called as the synthesis phase. Also called as the synthesis phase. All of you remember that? All of you will remember these 2 points, students. Hidden line of NCERT. Don't forget all this. They can ask you a question from anywhere. Okay. Now, the replicated chromosomes, that is the DNA, are then distributed to daughter nuclei by a complex series of events during the cell division. That is basically your M phase. The entire M phase. This is basically the M phase they are talking about. See, the replicated chromosomes, after S phase we have the G2, then we have the M phase, mitosis, right? Are then distributed to the daughter nuclei by a complex series. What is the series they're talking about? Not your Netflix series. They're talking about metaphase, anaphase, prophase, telophase. That is the series they're talking about here, not Netflix series. Of events during a cell division. These events are themselves under genetic control. That is all of your PMAT, prophase, metaphase, anaphase, telophase. All the following phases are under genetic control. Do you realize we decoded the paragraph? So students, this is the beauty of line by line of NCERT. This is where you understand every single line and you do not miss out any hidden line of NCERT, and you can answer any single question. How do I know that? I'll prove it to you. Watch the video till the very end of the session. We have pyqs, and you will realize whatever you are studying right now, every single point here can be asked in the form of a question, and you will solve this question towards the end of the video. Okay, you will be solving all those questions, not me. Okay, next paragraph. Students, any doubt in this paragraph? Cell division? I believe there should be no confusion here. Now students, now phases of cell cycle. What are the phases of cell cycle? Now students, if you want to broadly classify the cell cycle, it can be classified into two categories, broadly classifying. One is called as the interphase, other one is called as the M phase. Listen to me, understand. We go diagram wise we'll learn. Students, can you see this part here, the light blue part? The light blue part, this entire thing. This entire light blue part is called as your interphase. It is called as interphase. Okay. Now can you see this small black color portion? This small black color portion is called as the M phase. It is called as the M phase. Understanding? Students, is there any disturbance in the audio? Let me know that right now. Any disturbance in the audio? Sound check, check. Any disturbance? Is my voice not clear? Huh? Clear? Yes? No. Where were we? So I was telling you about the differences. There are two types of entire cell cycle can be divided into two categories. Entire cell cycle can be divided into your interphase as well as your M phase, Now students, remember, this interphase can also be further divided. This interphase can also be further divided into three categories. That is G1, S and G2. G1, S, G2. Voice is clear now? Don't lie, don't trick me and all. Don't lie, okay? Clearly audible, no? Nice. Okay. So G1, S, G2. That is the interphase division. Okay. This M phase also I'll tell you later on. We'll learn eventually. Okay. Now students, where were we? Let's come back to this paragraph. A typical eukaryotic cell. Now this point is also important. Why? Because in this entire chapter, we will be discussing about eukaryotic cell and not prokaryotic cell. Prokaryotic, we kicked it out, pushed it out. We'll be only talking about your eukaryotic cell in this particular class. Okay, remember that point. Is illustrated by a human cell in a culture. Now these cells divide once in approximately 24 hours. Very important line. That is the time required for division. Time required for division for a RBC cell. Right? Time required for a human cell. It's given RBC? No, it's just given human cell. Let's write human cell only. Human cell. Time required for a human cell to divide is going to be 24 hours. 24 hours. Okay? Now, 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. Again, very important for yeast. The entire cell cycle. For yeast, it's for around 90 minutes. Now, one more thing you should remember is the bacteria also. They might ask, but it is there in different chapters. Bacteria are approximately 20 minutes. Okay, 20 minutes. Now, the cell cycle is divided into two basic phases, interphase as well as the M phase. The meaning of M here is mitosis phase. All of you will remember this? Yes or no in chat? Any doubt here? Quickly ask me. Any doubt in this paragraph? Such an easy chapter. Why to worry? Why to cry? Easy chapter. Now students, again, this is what I was telling you. This is the end. This is the entire interphase. Interphase can be divided into three categories: G1, S, G2. This is the M phase, which I'll discuss later on. This is the entire M phase, which includes prophase, metaphase, anaphase, telophase, and cytokinesis. Done. Cytokinesis. Now students, let's read what is given here. The M phase represents the phase when the actual cell division or mitosis occurs. Very important line. What is M phase? In which the cell division or mitosis occur. That is, in S phase, there is a division of DNA content. In S phase, here students, in the S phase, there is duplication of DNA. Now we are DNA is duplicated here. DNA is duplicated. Now, for example, this is one cell. This is one cell. Now, within this one cell, students, within this one cell, there is this one DNA was there. Okay. Now, this DNA has duplicated now. This DNA has duplicated. This is just DNA duplication. What is the meaning of cell division? What is the meaning of cell division? Meaning of cell division is that this one cell will divide into two cells. This one cell will divide into two cells, and when it is dividing, these two DNA, different two DNA, one will go here and one will go here. Understood? Understood all of you? Deepika? This is the meaning of cell division. This is basically DNA replication. This is DNA duplication. DNA is becoming doubling here, doubling of DNA. This is cell division. Cell is dividing into one parent cell dividing into two daughter cells, and DNA is sent to each one of them. And this part happens in the M phase. This entire cell division happens in the M phase. Understand that. That concept is very important, very critical concept it is. Difference between DNA duplication and cell division. All of you understood? The difference between DNA duplication and cell division. This is called as cell division. This is DNA duplication. And this DNA duplication takes place where? DNA duplication happens in the S phase. This cell division happens in the M phase. Understanding? Tricky, no? Very easy, but it's very easy. Understood? Now students, now you need to understand one thing here. You need to understand one simple concept. I'll teach you a very simple concept now. Okay. How many months has it been that you are preparing for NEET examination? Tell me. How many months has it been since you are preparing for examination, NEET examination? Be honest in my class. How many months has it been since you are preparing for your examination, NEET examination? Let's say some of you 1 year, some of you 2 years. It can increase also. It can increase also. Do you realize you're working really hard for a year or 2 years, for 1 year or 2 years to write that one examination of 3 hours 20 minutes? I repeat again. You're working hard for 1 year, 2 years or 3 years sometimes to write that one examination of 3 hours 20 minutes. This long preparation time is there, right? This long preparation time is similar to your interface. It is similar to your interface. All of you right now are in interface. All of you right now are in interface now. That one day in examination, the NEET day examination, where you sit down and write the paper for 3 hours 20 minutes, that is going to be M phase. That is going to be the M phase. So what does it tell you? It tells you that interface is very long. Interface is very long. M phase is very short. Did all of you understand the concept? That is very important to understand also. Interface is very long process. Like how you're preparing right now. All of you are interface right now. If I ask you next time, what phase are you in? You're supposed to say, yes sir, I'm in interface right now. All of you are in interface. That one day exam you write right, that is going to be your M phase. Understanding? Yes. All of you, it's very easy concepts. Understand? Okay. Now students, let's come back to this. The M phase represents the phase when the actual cell division, all of you understand? The actual cell division is this, or mitosis occurs. And the interface represents the phase between the two successive M phase. Very simple to understand. Very simple to understand. See here. This is M phase. This M phase is between one cycle. Let me write it here only. This is going to be one interface. After this interface, there is going to be. After this interface, there is going to be a M phase. After this M phase, again there is going to be a interface. Again there is going to be a interface. So what is M phase, students? What is M phase according to your NCERT? It is the interface represents the phase between interface represents between the two M phases. See again, what will be here now? Here we'll have the M phase. Above that we'll have the interface. Now can I tell interface represents the phase between the two. It is a phase between the two M phases. That is the meaning of that line. Okay, understood? That is the meaning of that line. Now, it is significant to note that in 24 hours average duration of cell cycle of a human cell, the cell division properly lasts for only about an hour. What is the meaning of that line? Understand. I'll erase this. Students, I'll erase this part. Understand that line. Very beautiful line given by your NCERT. Understand this. Out of the 24 hours, there are two categories obviously. One is the M phase, other one is the interface. Other one interface. Out of the 24 hours, just 1 hour it is in the M phase. Just 1 hour it is in the M phase. Now how many hours are missing then? 23 hours. 23 hours it will be in interface. And remember I told you that time. Interface, all of your interface, which is the longest time. It is the longest time. Understanding? Understood? Now it also says that this 23 hours can I also write as 95%, and can I write this 1 hour as 5%? This 1 hour as 5%. So for a human cell, it requires 24 hours to divide. In that, M phase is just for 1 hour, interphase is for 23 hours, which is roughly around 95%. This is roughly around 5%. Understood? All of my students understood. I know. I have the confidence in all of you. All of you understood it because I am teaching in a very basic manner. Right? Now, let's read the line again. It is significant to note that in 24 hours, average duration of cell cycle of a human cell, cell division properly lasts for only about an hour. It talks about cell division. What is the meaning of cell division? Meaning of cell division is that the M phase. And M phase is only for 1 hour. About an hour. The interphase lasts for more than 95% of during of cell cycle. It is for 95%, which is roughly around 23 hours. And that is for interphase. Done. That's all. That's all it is. Such an easy concept. Understand now. Next line. The M phase starts with the nuclear division. Again, very important concept again. How does the M phase starts? M phase is divided into two categories. Can anyone tell me what is this chirokinesis? Cytokinesis. See, if you are all of you passed class 10th, no. All of you passed class 10th. In 10th, I teach you this. In 10th, you would have learned this. Chiro means nucleus. Kinesis means division. Division of nucleus is called as chirokinesis. Division of cytoplasm is going to be cytokinesis. Understand that. Division of cytoplasm and division of nucleus. Okay. The M phase starts with a nuclear division corresponding to the separation of daughter chromosome called as chirokinesis, and usually ends with division of cytoplasm, cytokinesis. Nuclear division, cytoplasmic division. The interphase, though called the resting phase. Tell me, students, I told all of you are in resting phase. Agree with that? I told all of you are in the resting phase right now. But are you resting actually? Tell me. Are you simply chilling in the night? No. Exactly at 6 p.m. you are in my class. You are learning something. Are you simply sitting quiet? No. You are writing down some notes. You are solving pyqs. You are solving DPPs. You are doing mock test. So in this entire 1 year journey of your NEET preparation, are you simply sitting quiet? No, you are not sitting quiet. You are hard work. Right? You are working hard. So cell is also working hard. So in this interface, the cell is not resting, the cell is not keeping quiet. The cell is working hard, like all of you. I told you, all of your interface, you are supposed to work hard, right? It's given here. See, the interface, though called as the resting phase, is the time during which the cell is preparing for the division by undergoing both cell growth and DNA replication in an orderly manner. Like how you are preparing right now for NEET examination, cell is also preparing for the M phase. It is preparing for the M phase. But how is it preparing? You are solving questions, you are doing revisions. But what is this revision for the cell? What is the preparation for the cell? The preparation for the cell is nothing but cell growth as well as DNA replication. The most two important points of this paragraph. How is the cell preparing? The cell is preparing in the interface by cell growth, which happens in G1 and G2, DNA replication, which happens in the S phase. Done. In an orderly manner. The interface is divided into three further categories. I told you already. The G1 also called as gap 1, S phase called as synthesis phase, G2 called as gap 2 phase. Any doubt till now in this paragraph? Quickly go. Any doubt in this paragraph? And I'll find my bottle. I don't know where I kept it. Oh, I got it. Students, all of you drink some water. All of you drink some water now. Drinking is very important. All of you need to drink water. Keep rehydrating yourself. Done. Understood, students? Can we go to the next concept now? Can we start with the next concept? Yes or no? Notification sometimes can, not come. What will happen? Notification does not come. 6 o'clock some class starts in the Vedantu NEET English channel. All of you are here at 6 o'clock, right? All of you here to learn. So I am happy about it. Okay, students, quickly smash that like button. And all of you watching recorded also, please smash that like button. What is this? Where is the energy? All of you are like, oh, sir, NCERT line by line. We will kill it. Where is the energy now? Show the energy. Okay. Hello, Gautam. Gautam is back. Gautam is one of my old students. And let me tell you, Gautam, this year, he'll definitely get into a medical college. Right, Gautam? Gautam is my old student from last year. Right? I remember Gautam wrote the examination, he was getting more than 650 or something, he told me. 650 plus. Right? Nice. Now students, let's understand what is happening in G1, S and G2. G1, S and G2. Ready? I can understand Telugu. You think I can't understand Telugu? Huh? You think I can't understand Kannada? You think I can't understand Tamil? All the three languages are sister languages. And it's very interrelated. I can read Kannada, I can understand Tamil and Telugu also. It's very easy. It's not rocket science and all. Right? Nice. Now students, let's understand what is G1, S and G2 now. Okay. What is G1, S and G2? What exactly is happening in all the three? G1, S and G2. Understand this very well. First, understand what is happening in G1. Okay. Let me write some few points for you. Students, write down this point now. Don't be lazy, write down these points. What exactly happening in the G1? What exactly happening in G1 right now? Okay. G1 is the first stage. It says 1 year only, so it is the first stage, right? It's actually the longest phase. It is actually the longest phase in the cell cycle. Longest phase in the cell cycle. Okay, longest phase in the cell cycle, or can I say longest phase in the interface. Longest phase in the interface. Done. Now what exactly happens in the G1 phase? What exactly happens? Now the cell is preparing. Right now. It is ready. It is preparing. Okay. So how does it prepare? How can the cell prepare? Tell me. Can you guess? Obviously, cell needs to make its weapon. The weapons are proteins and RNA. That's all. So what exactly happens is active synthesis of student. This is a PYQ, students. Let me tell you. This question has been asked for PYQ. This line is a PYQ. Multiple PYQs. Active synthesis of RNA and protein. Very important. RNA as well as protein. Most organelles duplicate here. Most organelles duplicate here. Most organelles duplicate here. But is there a DNA replication here? Is there DNA replication? No. No, absolutely no. No DNA replication. No DNA replication. Done. Only the following points which you need to know. How many? A 1, a 2, a 3, a 4. 4 points. That's all. Done. This is the G1. Still the Diksha ma'am joke is going on, no? Still the Diksha ma'am joke is going on. Yes, my name is Diksha ma'am. Huh? Beta, what to do now? Beta. Okay. These are the 4 points which you need to know about G1 phase. That's all. Now, what about S phase? What about the S phase, students? S phase, let me tell you, there are only 2 main points. Let me tell you. In S phase, what exactly happens? Friends, in G1 phase, this is how the chromosome looks like. Okay. This is the G1 phase. What exactly happens in G2 phase? In G2 phase, students, tell me, this is 1 chromosome or 2 chromosome? Tell me, this is 1 chromosome or 2 chromosomes? Only 1 chromosome. Only 1 chromosome. Okay. Only 1 chromosome. Now, this 1 chromosome, if it looks like this, is this still 1 chromosome or 2 chromosome? Tell me. Is this 1 chromosome or 2 chromosome? Tell me now. If you tell 2 chromosomes, I'll hit you now. It is only 1 chromosome again. It is still only 1 chromosome, but it has 2 sister chromatids. 2 sister chromatids. It has the sister chromatids now. Understand that. And this exactly happens. This is, here we had only 1 DNA. Yeah, we have 1 DNA. We had 1 DNA. Okay. Now this DNA becomes 2 now. This DNA becomes basically 2. This DNA becomes 2. Okay. Formation of sister chromatids. So what exactly happened here? The DNA content from 2C. This is called as 2C. From 2C, the DNA content became 4C. The DNA content became 4C. Now I will ask you one more question. Is there increase in number of chromosome? This also was 1 chromosome. This also is 1 more chromosome. So answer is no. There is no increase in number of chromosome. There is no increase in number of chromosomes. No. Number of chromosomes will remain the same. Number of chromosomes will remain the same. Only the DNA content is increasing. That's all. Only the DNA content is increasing. DNA content is increasing. DNA content is increasing. Understood? Yep. That is basically, this is nothing but DNA replication. DNA replication. This is your DNA replication. Apart from that, there is also centrioles. All of you remember centrioles? Tell me how many centrioles are there? 1, 2, 3, 4, 5. How many are there? They are in a cartwheel-like structure, together called a centrosome. How many centrioles are there? Oi, tell me how many centrioles are there? Fast. There are 2 centrioles, right? There are 2 centrioles in 1 centrosome. In 1 centrosome, there are 2 centrioles. So centrioles also duplicate here. Centrioles duplicate. Centriole duplicates in the cytoplasm. That is very important. It divides in the cytoplasm. Also, there is histone protein synthesis. Extra point this is for all my students. Histone protein synthesis. That's all. So easy. Done. All of you are very smart now. All of you are becoming smart day by day. Okay. Now what happens in G2? Everything is happening in G1 and S. Is something left in G2? Yes, something is left in G2. Yes, something is left in G2. That is duplication of mitochondria, the powerhouse of the cell. Duplication of mitochondria. Duplication of mitochondria happens. Yes, and the spindle proteins. You need spindle fibers, right? The spindle proteins also. Spindle proteins. And also some of the RNA synthesis of even some of the RNA also happens here. RNA protein also happens. Some of them, not major. Majority of RNA and proteins happens in the G1. Okay, happens in G1. Oh, genius. G for genius. All of you wait. Parle G. What to do? Okay. Let's read now. Let's read. Quickly read. The G1 phase corresponds to the interval between mitosis and the initiation of DNA replication. What is the meaning of this line? Let's draw this. Let's draw this. What is the meaning of this line? Let's draw this. DNA phase corresponds to the interval between mitosis and initiation of DNA replication. Mitosis and initiation of DNA replication. That is, I told you, initially I told you, we have interphase, up we have M phase, below also we have the M phase. Okay. Interphase is divided into G1, S, G2. So technically speaking, your G1 phase, is between what? The G1 phase is between mitosis on the top side and initiation of DNA replication. That is going to be your S phase. So G1 phase is between M phase and S phase. That's all. So once you write everything, it becomes very easy to quantify also. All of you will go to your brain. Okay, understanding or not understanding? Gone, already gone out of your head. Understand? That is the meaning of this line here. So this is the importance. This is the importance of line to line of NCERT. And when you draw it like this, it becomes easy to understand also. Right? So G1 phase corresponds to the interval between mitosis, M phase, and initiation of DNA replication. That is the S phase. During the G1 phase, the cell metabolically active and continuously grow but does not replicate. Very important line. Does not replicate its DNA. Quickly, ask you a question now. True or false? Okay. True or false question. Focus. In G1 phase, DNA replication takes place. True or false? Quickly. In G1 phase, DNA replication takes place. True or false? And don't think I'm asking easy question. This is a NEET PYQ also. This is a NEET PYQ. Tell me. In G1 phase, DNA replication happens. True or false? Go. Next, S or synthesis phase marks the period during which DNA synthesis or replication takes place. That's what we learned here. DNA replication, S phase DNA replication. Okay, replication. During this time, the amount of DNA per cell doubles. DNA per cell doubles. Deepika, not true, ma. How can it be true? I told you 100 times. I'm telling you, Deepika, understand. DNA replication will not happen in G1. DNA replication will happen in only S. Understand? Think. It happens only in the S. Okay. In G1 it does not happen. I wrote also no here. Forgot. See, no DNA replication. G1. Understood now? It's a PYQ. And don't think I'm not looking at the chat. I'm looking at the chat also. So don't tell lies also here and there. If the initial amount of DNA denoted to be 2C, then it increases to 4C. That is 2C to 4C. 2C to 4C. However, there is no increase in chromosome number. Okay. Simple question. In S phase, in the S phase of cell cycle, how many chromosomes are increasing? Into 2, into 3, no increase at all. What is the answer? Tell me. Tell me. Simple question. In the S phase of interphase or cell cycle, how many number of chromosomes are increasing? Into 2, into 4, or no increase at all? No increase at all. If the cell had diploid or 2 in number in the G1, even after S phase, the number of chromosomes remain the same, that is 2n. So what exactly is happening? Understand this. Understand this point here. Very important also. In G1 phase, it was 2n, 2c. 2n, 2c. In S phase, what happens? N is the number of chromosomes, C is the DNA content. It becomes 2n. 2n remain the same, but this 4 or 2c will become 4c. Understood? Understood? Only the DNA content is increasing. Only the DNA content is doubling, but number of chromosomes are going to remain the same. Very simple concept, yet many questions have come from here. Many questions have come from here. So if you do not want to make the mistake, if you do not want to make any kind of stupid mistake, write down in your book right now. And once you write down, tell me done in the chat. Don't tell done in the chat. I want to see the done. That is 2 and 2C2, 4 and 4C. Everyone, NCERT right now only. Later if you write, you'll forget it off. I know, pakka promise you'll forget it off. Next. In animal cells, during the S phase, the DNA replication begins in the nucleus. All of you know nucleus it happens. And centrioles duplicate. I told you, centrioles duplicate in the cytoplasm. Now I'll ask you a question. Simple question again. Centrioles duplicate in which phase of cell cycle? In which phase of cell cycle? G1, S, G2 or M phase? Question is very simple. Centrioles duplicate in which phase of cell cycle? G1, S, G2, M. Answer is S phase. Answer is going to be S phase. During S phase, the DNA replication begins in the nucleus and the centrioles duplicate in the cytoplasm. Simple question. During the G2 phase, protein are synthesized. I told you, protein are also synthesized in the preparation of mitosis while cell growth continues. Cell growth also continues. Very beautiful. Done. Any doubt in this paragraph, quickly ask me. Any doubt in this paragraph, quickly ask me, students. Any doubt here? Any doubt? Anyone? Anyone? Any doubt? Cell cycle. We will kill it today. We'll kill it today. Easy concept. After today's class, we should never cry and come to me telling that, sir, I'm not understanding cell cycle. This is it. This is the chapter which you want. Okay? This is the chapter which you have to put in full stop here only. Now, this I wanted to give as homework question, but this will do in the end. Remind me, this will do in the end. Okay, very easy, but we'll do in the end. Now some of you are asking me, sir. Some of you are asking me, sir, there is something called a G0. G motor. What is it? G0? G0. Divya is asking, please smash the like button. All of you listen to Divya. So G2 phase tells, sir, in G2 phase nothing is happening. Listen. In G2 phase, what is happening? In G2 phase, nothing is happening. Duplication of mitochondria, spindle proteins, and mostly, don't tell RNA. RNA usually happens there only. Protein synthesis is happening, that's all. Protein synthesis. G2, protein synthesis. NCERT is not giving you anything, but let me tell you. Okay, that's all. During the G2 phase, proteins are synthesized in preparation of mitosis. That's all. Now, so tell something about G. It's coming. Have some patience in life. What is the G0 then? What is the G0? Very simple example. Very simple. G0. Some cells are fed up in life. They're depressed. Full depression. Full depression in their life. Okay, some cells. In some cells, what happens is, after the G1 phase, and they are like, I can't do anymore. So after G1 phase, students, some cells after G1 phase, here, some cells after G1 phase, they are exiting the cell cycle. After G1, that is very important, it's a PYQ. It is a PYQ. After G1 phase, they are exiting the cell cycle. They are exiting the cell cycle. Yes, very depression. Too much of depression they have. They are like, I can't do anymore. They are exiting after G1 phase. Exiting after G1 phase. Can you see here? Some cells in adult animals do not appear to exhibit division. Hard cell does not divide any further. And many other cells divide only occasionally as needed to replace that has been lost because of injury or cell death. That is, whenever there is requirement, they divide. There is no requirement, they exit. Okay. Sorry, don't scare me like that. You are hearing some noise. I am not hearing any noise. Don't scare me like that. Okay. Students, what is something wrong? Is something wrong with the chat? Huh? I am not hearing any noise. I don't know what you are hearing. Some extra you are hearing from somewhere you are hearing. Oh, what is this sound? Shaking sound. What is this shaking sound? Okay, 1 second. Let me keep this mic very close to each other. Listen to me. Now? There's no noise now, right? Any sound right now? Sound is shaking. Physics Physics RIP. RIP physics. RIP physics now. Sound is shaking. Okay. Now, these cells do not divide further and exit the G1 phase to enter an inactive stage called as quiescent or quiescent stage. So, quiescent stage or G0 phase. Okay, G0 phase is basically quiescent stage where the cell is exiting G1 phase and it is entering the G0 phase. Understood? That is called as G0 phase is an inactive phase. It is a inactive phase where the cell does not divide. Okay. No, it's proper now, right? No noise, right? All of you some ghost stories are telling me now in the middle of a class. Okay. Now these cells do not divide further and exit G1 phase to enter an inactive stage called as quiescent or G0 phase of the cell cycle. The cells in this stage, very important line, cells in this stage remain metabolically active. So question will come. Okay. In G0 phase, the cells are inactive and also metabolically inactive. True or false? In G0 phase, the cells are inactive and also metabolically inactive. True or false? They are not going to be. They are going to be metabolically active. They cannot be metabolically inactive. So it is a false statement. Why? Because cells in this stage remain metabolically active, but no longer proliferate unless called upon. So to do depending upon the requirement of the organism. So whenever there is requirement, there will be division. If there is no requirement, no division. Okay? Understood. Now students, last paragraph. In animals, mitotic cell division is only seen in diploid somatic cells. What is the meaning here? Its meaning is that mitosis is seen in the diploid cells. That is, if a cell is diploid, 2N, it will undergo mitosis and will produce 2 cells. So both the cells are going to be 2N only. Okay, 2N to 2N. That is the meaning here. However, there are few exceptions to this where a haploid cell divides by mitosis. There are some exceptions in which a haploid cell is dividing into 2 cells. A haploid cell is dividing into 2 more haploid cells. Example in the case of male honeybee, that is your drone. That is going to be your drone. Okay, drone. Okay, drone. Against this, the plant also show mitotic divisions in both haploid and diploid cells. That is your algae. All of you know the life cycle of algae and bryophytes, right? In which a haploid cell can give rise to a haploid. Okay, haploid. From your recollection, that is plant kingdom. You have learned in plant kingdom. Okay, identify the plants, that is bryophytes and pteridophytes. Done? Any doubt in this paragraph? We are done with cell cycle actually. That's all the concept of cell cycle. Okay? Done or not done? Yes? One second students, let me just check. Is there any problem? No problem. And students, how many of you are aware that tomorrow there is a class? How many of you are aware that tomorrow we have a class at 12 p.m.? How many of you know that? Tomorrow 12 p.m. we have a class on genetics. So please attend that. All of you are crying to me, sir, please make one confusion. Please make one genetics question. I am making it tomorrow. So you better all of you attend it. Otherwise I'll make it private. Now I'll make it private. Okay. So tomorrow we have a class of numericals. All of you have been requesting me, sir, please bring numericals. Okay. And go smash that like button there quickly. Okay. Now students, with that being said, can we start with the M phase now? Nicely 1 hour we did. Okay. Can we start with the M phase now? Show some energy in the chat now. Can we start with the M phase now? Is the sound proper now? Check once. Is the sound proper or not proper? I think that is the AC sound. What you're getting is the AC sound. Let me stop the AC. Sound will go eventually. Don't worry. Can we start now? Drink some water, all of you. Drink some water. Very important to drink some water. Done? Can we start now? Now, let's start with the M phase now. Let's start with the M phase. Very important, very easy. In next 45 minutes, we'll finish it off. 45 minutes, we'll finish it off. Questions are there. Don't worry, questions are there. After 45 minutes, after MC students, I'll show you. After the M phase, we'll solve questions. What's it? See, all PYQs. Do you see? All the PYQs will be doing in today's class only. All the PYQs will be doing in today's class only. So don't worry. PYQs are coming up. Okay? Where will be now? Where will be? Okay, we are here somewhere. We'll starting here. Okay, we'll start it here. Now students, understand. I told you the entire cell cycle is divided into two categories, right? Entire cell cycle is divided into two categories. That is your interphase as well as the M phase. That is going to be interphase as well as the M phase. Interphase as well as the M phase. Interphase divided into three categories: G1, S and G2. G1, S and G2. This M phase is further divided into two categories. M phase is further divided into two categories: chirokinesis and cytokinesis, as well as cytokinesis. Okay. This chirokinesis is further divided into four stages. That is PMAT. M-A-T. PMAT. That is prophase, metaphase, anaphase, as well as telophase. Now you might ask me, sir, is it important to learn the order also? Is it important to learn the order prophase, metaphase, anaphase, telophase? Yes, it is important to learn the order also. Because there are PYQs based on the order also. Prophase, metaphase, anaphase, telophase. There are questions based on the order. PMAT. This is called as PMAT. Okay. P-M-A-T. PMAT. All of you remember PMAT. Prophase, metaphase, anaphase, telophase. Done. This is what you'll be learning in today's class. You'll be learning in today's class. Okay. Now. This is the most dramatic period of the cell cycle. Full, like teenage, like all your teenagers know. Full drama. Like Big Boss house also. It is like Big Boss house. Involving a major reorganization of virtually all components of the cell. That is, Golgi will disappear. All the Golgi will come here. Endoless reticulum will come there. Entire mix and match is happening. Okay. Since the number of chromosomes in the parent and progeny is same, parent is 2n, offspring is also 2n. 2 and 2n is maintained, right? It is also called as equational division. Meaning of equal is what? Equal. Equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division, chirokinesis, it is very essential to understand that the cell division is progressively processed and very clear-cut lines cannot be drawn. So basically what we learn, prophase, metaphase, anaphase, it is not always the case. Sometimes here and there mistakes can happen. Okay. Now, cannot be drawn between the various stages. Chirokinesis involves following: PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. Any doubt, quickly ask me. Students, how many of you remember Telegram? Tell me honestly. How many of you remember Telegram? That is, this is my Telegram, student. This is my Telegram. Basuraj sir, biology. On this Telegram, when a few days back, few weeks back, I uploaded an image of my sectioning, under a microscope, which showed various phases of mitosis. Yes. In my Telegram, I added an image of a cell under the microscope, which showed multiple stages of your prophase, metaphase, anaphase, telophase. So if you're not in my Telegram, quickly go join it and see that for yourself. It is an actual image taken by me, not from internet and all. No copyright. Okay. Now, we'll understand prophase now. We'll understand prophase. Very easy to understand. Students, can I draw and explain first? Can we draw and explain, understand this prophase? I will tell once. I'll tell you. Understand with me first. Understand with me. Prophase, let me write for you first, and then we'll understand what is written in NCERT before we go to the NCERT. Okay. First, you need to understand prophase. Okay. Prophase. What you need to understand in prophase? First of all, you need to understand it is the longest phase of M cycle. It is going to be the longest phase in M phase. Longest phase of M cycle. Longest phase of M cycle. Of M phase. Okay, longest phase of M phase. Now, what exactly happens here? In the case of your prophase, there is initiation of condensation of chromosome material. Initiation of chromosomal initiation of chromosomal condensation initiation of chromosomes condensation of chromosomes. Okay, now if you can divide the entire prophase, you can divide the entire prophase into early prophase and late prophase. Let's divide the entire prophase into two categories. Let's divide the entire prophase into two separate categories. I want ultimate focus right now. Ultimate focus, do not see here and there now. Write down this. Once you write down, you will stuck in your head. Okay. Now, entire prophase can be divided into two categories. Early and late. Early and late. Early prophase, early phase, early prophase, as well as late prophase. Late prophase. Understand this point very well. Early prophase as well as late prophase. Now, what exactly happens in early prophase? First of all, in early prophase there is condensation of chromosome. There is condensation of chromosomes. Condensation of chromosomes there. There is condensation of chromosomes. Okay. Now, apart from condensation of chromosomes, there is something called as movement of centrosome. There is movement of centrosomes. Movement of centrosomes. Understand? Handwriting is looking nice now suddenly. Students, handwriting is looking so nice suddenly. I don't know what happened. Okay. Movement of centrosomes. Understand this. Okay. Movement of centrosomes. I will draw and show you. Okay. This is a cell. This is one cell here. Okay. Imagine this is a cell. Now, what is happening, students? First of all, first point. What is the first point? Condensation of chromosomes. If this is the entire nucleus, this is the nucleus. Within the nucleus, what is happening? Within the nucleus, there is condensation of chromosomes. Chromosomes are getting condensed. Chromosomes are getting condensed here. Chromosomes are getting condensed. Apart from chromosomes are getting condensed, what exactly is happening? Movement of centrosome. Can anyone remind me what is a centrosome? All of you remember from the cell chapter. Centrosome it has the outer layer called as amorphous pericentriole or amorphous structure, amorphous pentricular material. It is made up of two centrioles. Which are in cartwheel structure. Cartwheel structure, all of you remember? Cartwheel structure, right? Cartwheel structure. That is what is happening here, first of all. That is what is happening. The arrangement of the movement of two centrioles to the two opposite poles. Done. The movement of centrioles or centrosome into the opposite poles. Done. Movement of centrosome is happening. Now, students, one big concept is coming now to all of you. You might have heard about it, it might be new to you, but understand this point. How many of you remember I taught you something called as cytoskeleton. How many of you remember cytoskeleton? If you have not watched the Cell, the Unit of Life, this chapter is very difficult to understand. How many of you remember the cytoskeleton? Especially the something called as microtubules. 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 You should remember microtubules, because there are two types of microtubules here. There are two types of microtubules. Two types of microtubules. Two types of microtubules are here. Two types. Understand? Okay. Two types of microtubules. First one is called as asters. First one is called as asters. The second one is called as spindle fiber. Spindle fibers. Asters and spindle fibers. Now show me, sir, in the diagram you show me now, sir. Students, understand from this region right here. You see, here in the back side, we have the asters. We have the asters. The main function of asters is almost anchorage. It is going to be anchorage. Okay, anchorage. Here we have aster formation. Asters in the bottom. Asters. And what about spindle fibers? Spindle fibers. Spindle fibers also there. From here, spindle fibers are rising. From front, spindle fibers are arising. Both are a type of what? Both are a type of microtubules. Both are a type of microtubules. Did all of you understand the concept? Yes or no? First of all, tell me. In early prophase, there is condensation of chromosome, movement of centrosome to the opposite poles. Two types of microtubules. Asters here and spindle fibers coming out like this. And one more thing you need to remember is the together, that is asters plus spindle fibers. Asters plus spindle fibers together, they are called as mitotic apparatus. They are called as mitotic apparatus. Together they are called as mitotic. They are called as mitotic apparatus. Mitotic apparatus. Beautiful concept. Done. Now students, if you look at what exactly happens in late prophase then. What exactly happens in the late prophase then? What exactly happens in the late prophase then? Late prophase. Understand. Understand this. What exactly happens in the late prophase then? In late prophase. In the case of late prophase, the different organelles. Understand. Shivashri, wait. Have some patience in life. Have some patience in life. Don't spam. In the late prophase, entire nuclear membrane, endoplasmic reticulum, right? Nuclear membrane, endoplasmic reticulum, as well as your Golgi complex and nucleosome. Golgi complex as well as your nucleolus disappears. Understood? Understood? Understood all of you? Yes or no? Quickly in the chat. Did all of you understand this point? Yes or no? Quickly. Now I will tell you one more part here. This is an extra concept you need to understand. Spindle fibers are present both in the animals and plants. Animals and plants. But asters are only found in the case of animal cells. Understood? Shramshi, did you get your understood? Understood? AC is off. I am sweating now. Students, I'll recap one more time for all of you. Okay. Prophase, longest phase of the M phase, initiation of condensation of chromosomes, early and late. In early, condensation of chromosomes, movement of centrosomes. Two types of microtubules, asters and spindle fiber. Asters here, spindle fiber, this ones. Together they are called as mitotic apparatus. Asters only in the case of animals, spindle fibers both in animals as well as plants. In late prophase, what happens? In late prophase, nuclear membrane, ER, Golgi complex, nucleolus, everything disappears. One oven it is becoming now, oven. Full hard it is becoming. Now, prophase, which is the first phase in the eukaryokinesis of mitosis, following the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecule are formed, are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. Did we read that point? Initiation of condensation of chromosomal material. Okay. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosomes, which had undergone duplication during the S phase of interphase, remember S phase of interphase, okay, S of interphase, now begin to move toward the opposite poles in the cell. Okay, the completion of prophase can thus be marked by the following characteristic events. What are the events? Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes seem to be composed of two chromatids attached together at the centromere. Here, this is basically talking about. Okay, centrosomes which are undergone duplication during the interphase now begin to move towards the opposite poles. Each centrosome radiating out microtubules called as asters. I told you, asters are there. The two asters together with spindle fibers form the mitotic apparatus. Now, how many asters did we make? One aster this side, second aster this side. Two asters. There are two asters. Okay, two asters. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus, L was missing, yes, nucleolus and nuclear envelope. Everything disappears by the end of, this is your late prophase. This is nothing but late prophase. Done. So please take meiosis, discuss more numerical problems. Done. We'll do it. Krish, what is so worry? We will do numericals on mitosis also. Do you want to do? We'll do numericals on mitosis, meiosis, both if you want. Don't take tension in life. Okay. Now, see, early prophase, late prophase. Nuclear envelope is disappearing, complete condensation of chromosomes and endoplasmic reticulum, Golgi complex, everything is missing. Late prophase. Done. Now, metaphase. Understand metaphase now. Oh. Friends, are you able to see this entire thing is not visible or not visible? Visible? Yes, visible? Visible, right? So, any doubt in prophase now? Ask me. Any doubt in prophase? Any doubt in prophase? Quickly ask me. Go. Any doubt in prophase? No doubt? No doubt? Perfect. If there is no doubt, can I explain mitosis first? Can I explain the metaphase with the help of diagram first? Yes? Can we? Shall we? Before reading, let's understand first of all. Very important to understand, no? What exactly is happening? Okay, what exactly is happening in the case of your mitosis, in the case of metaphase? What exactly is happening in the case of metaphase? Students, this is your entire cell. What do we have in the corners, in the opposite poles? The two centrioles. The centrosome. It is radiating out spindle fibers. Covered by the amorphous pericentriolar material. Okay. Now, in the case of metaphase, all the chromosomes align at the equatorial plate. All the chromosomes align at the equatorial plate. All the spindle fibers are attaching. Now, I want you to understand one simple concept here. I want you to understand a simple concept first of all, before we go ahead. Okay. This is the primary constriction. The primary constriction is called as your centromere. On this primary constriction, we have the two sister chromatids. Okay. On the sister chromatids, there is a proteinaceous layer. Called as kinetochore. Okay. There is a proteinaceous layer called as kinetochore. Okay. Kinetochore. This is a very important concept, PYQ also. Do you see the attachment of spindle fibers? The spindle fibers will never attach to the centromere. In fact, the spindle fibers will attach to the kinetochore. They'll attach to the kinetochore of centromere. All the spindle fibers will attach to the kinetochore. Okay, kinetochore. Now, what exactly happens in the metaphase? There is movement of chromosome. First of all, there is movement of chromosome. That is called as congregation. Movement of chromosomes. Yes, there is movement of chromosomes. Movement of chromosome is happening. All the chromosomes are moving towards the middle plate. This is called as metaphasic plate. This plate, can you see this pen? Green color one. This is called as metaphasic plate. Is called as metaphasic plate. The other name for this metaphasic plate is also called as equatorial plate. It is also called as equatorial plate. What is this? This is the spindle fiber. It's the spindle fiber. Okay. What is happening here? Spindle fiber attached to the kinetochore. Spindle fibers are attaching to the kinetochore. Now they can also ask you here more question. How many kinetochores are present? They can ask you a simple question. How many kinetochores are present per chromosome? What is the answer? Tell me. How many kinetochore are present per chromosome? You will tell me, sir, there are 2 kinetochores present per chromosome. Now, if there are two kinetochore per chromosome, how many will be there in a diploid cell? How many kinetochore do you find in a diploid cell? Diploid cell has 46 chromosomes. It has 46 chromosomes. So 46 into 2. What is the answer, quickly? Maths. Go. What is 46 into 2? Go. That is the answer for your, this is your metaphase. That is going to be 92 there. Okay, 92. Now one more thing, apart from that, there is best stage to study the chromosome morphology. This is the best stage to study chromosome morphology. Best stage to study chromosome morphology. Now you might ask me, sir, why? Why is it the best stage to study morphology? Because the complete condensation of chromosome is completed in the metaphase. Understood? The complete condensation happens in the metaphase. Condensation of chromosomes. Complete condensation of chromosomes happens in the metaphase. Very simple. This is 92, right? 92 chromatids are there. 92 chromatids are there. Clear? This is the metaphase story. Story of metaphase. Let's read what is given here now. The complete disintegration of nuclear envelope marks the start of second phase of the mitosis. Hence, the chromosomes are spread throughout the cytoplasm of the cell. Okay. By this stage, condensation of chromosome is complete. You see, condensation of chromosome is complete here, and they can be observed clearly under the microscope. That's why it's the best stage to study the morphology of your chromosomes. This is then this is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosomes is made up of two sister chromatids here. Two sister chromatids. How many? Two sister chromatids. Two sister chromatids, made up of two sister chromatids. Okay. Which are held together by a centromere, the primary constriction. Small disc-shaped structure at the surface of centromere called as kinetochore are also present. Disc-shaped kinetochore. These structures serve as a site for attachment of spindle fibers. Very important line. Formed by the spindle fibers to the chromosomes that move to the position of the center of the cell. Hence, metaphase is characterized by the all chromosomes coming to the lie at the equator with one chromatid of each chromosome connected by its kinetochore to the spindle fiber from one pole, and its sister chromatid connected to by the kinetochore to the spindle fibers for the opposite pole. What I'm trying to tell you here is this paragraph here. We drew this. One sister chromatid from one sister chromatid with the help of one kinetochore is attached to the one part of the centriole. Another sister chromatid with the help of a kinetochore is attached to the another opposite side of a centriole. So basically, the two different sister chromatids are attached to the two different poles. Did all of you understand that point? Yes or no, quickly in the chat. That is what is given in this entire part. Okay, that is characterized by all the chromosomes coming to the lie at the equator, one chromatid of each chromosome connected by a kinetochore to spindle fiber from one pole, and its sister chromatid connected by again a kinetochore to a spindle fiber from opposite pole. So opposite poles. Okay, opposite poles. The plane of alignment of the chromosomes at the metaphase is referred to as metaphasic plate. That plate is called as metaphasic plate. The key features of metaphase are spindle fibers attached to the kinetochore of the chromosomes. Done. Chromosomes are moved to the spindle equator, the middle one. Spindle equator, metaphasic plate, equatorial plate, same meaning, and get aligned to the metaphase through the spindle fibers to the both the poles. Done. Metaphase dusted. Done and dusted metaphase. Students, quickly smash that like button. All of you are not marking your attendance. And all of you, I realized one more thing. Okay. I was looking at the comments last time. And all of you are not putting the effort. We are putting so much of effort. Where is your effort? Huh? We should also comment sometimes. You should try commenting. Okay, magic will happen. I will reply this time. If you comment, I will reply also this time. Okay, show the love towards the comments, because it is the only way I will know that you are understanding, right? See, transition to the metaphase. This is the entire metaphase. Entire metaphase. Okay. Now, friends, can we do anaphase now? Can we do anaphase now? Again, anaphase is very easy to understand, yet the most important part of today's event. Can we start? Can we start, students? Can we start? Yes. Obviously, why will you say no? Learning is the most important thing, right? Now, understand anaphase. Anaphase is very important for you to understand the DNA content. Understand anaphase. What exactly is happening in the anaphase? Okay. Let's draw it. First, let's draw and understand. That is the best way to learn. Let's draw and understand first the entire anaphase. This is your drawing. Okay. Let's draw and understand. Okay. Now, this is the entire cell. In the middle, what do you have, students? In the middle, we have the metaphasic plate. Metaphasic plate in the center. Okay. In the corners, we have the centrioles. Centrioles. Here we have centrioles. Okay, centrioles is there. It is covered by a pericentriolar amorphous material. Amorphous pericentriolar material. Okay. Now, what is happening here to the chromosomes? Now, what is happening to the chromosomes? The chromosomes which were in the center, the two chromosomes which were in the center, did they look something like this? Yes. They looked something like this. In the middle, we had the centromere. So, what is happening in anaphase is, what exactly is happening in the anaphase is that in the middle, there is a breakup happening. This entire chromosome is splitting into half. The entire chromosome is splitting into half. Okay. The entire chromosome is splitting into half. That is, the centromere is splitting and the two sister chromatids are being pulled apart. That's all happening. Sad story, this is one. This is one sad story is happening here. We end up getting something like this. We end up getting some structure like this now. Done? That is what is happening here. Some sad story. That is, the sister chromatids are splitting up. And they are being pulled to the opposite poles. They are pulled to the opposite poles. They are being pulled to the opposite poles. They are being pulled. Okay. Okay. Let me draw it better. One heart looks like one heart. All the chromosomes are being pulled. All the chromosomes are being pulled apart. Yes, Pandu and Chamba separation. Full massive breakup is happening. Okay, massive breakup is happening. Okay. So what did we observe, students? What did we observe here? The first thing which you observed, first of all, this is the shortest phase. It's going to be the shortest phase. It is going to be the shortest phase. Oh. It's going to be the shortest phase. What is happening here, students? What is happening here? The sister chromatids are separating. The sister chromatids are separating. Sister chromatids are separating here, separating of sister chromatid. So separating of sister chromosomes or sister chromatids. Sister chromatids are being separated out. Okay. Now, I will teach you an important concept here now. Understand this point. Understand this point. Understand this extra point. Now, students, tell me in this cell here, what exactly happened here right now? The two sister chromatids being separated out. Okay, two sister chromatids being separated out. Now, can I call these daughter chromatids now? Since they are not together now, they are no more sisters, they became daughter. They are daughter chromatids. Now, these two daughter chromatids will act like, daughter chromosomes. They will act like daughter chromosomes in the future nuclei. These two daughter chromatids, these daughter chromatids will act like daughter chromosomes in the future nuclei. Understood? These two daughter chromatids are there, right? They will split open after cytokinesis happens. After cytokinesis happens, this will become one cell, this will become one cell. So each individual chromatid will act like a chromosome now. Tell me. Each individual chromatid, will it act like an entire chromosome now in the future? Yes, it will act like in the future. So right now tell me, how many chromatids are there in this particular cell? How many chromatids are there in this particular cell? How many chromatids are there? How many chromatids? So this is a diploid cell. This is a simple diploid cell in which we had 46 chromatids. 46 chromatids. Now, what exactly will happen, you say? All these 46 chromatids, students, all these 46 chromatids will act like 46 chromosomes. 46 chromosomes. So, students, in the anaphase, temporarily, in a temporary fashion, in anaphase, temporarily, in one cell, there will be 46 chromosomes. So, there will be, sorry, 92 chromatids, sorry, not 46, 92 chromatids. What am I saying? 92. 46 chromosomes, 92 chromatids. Right? There will be 92 chromatids, right? 92 chromatids. So, temporarily, there will be 92 chromosomes. 92 chromosomes, not 46. Understanding? This side will be 46 chromosomes, this side will be 46 chromosomes. So, in total, temporarily, in anaphase, there will be 92 chromosomes. Let that sink in. Let that sink in. If you want me to repeat again, ask me again. This is what the important concept here is. So, in the case of anaphase, temporarily there will be all the 92 chromatids will act like a chromosome, will consider like a chromosomes. They are called as daughter chromosomes, daughter chromatids. So, in the anaphase, temporarily there will be 92 chromosomes. There will be 92 chromosomes. Understanding? All of you? I want to answer from all of you quickly. Tell yes sir in the chat. All of you quickly. Yes sir in the chat. Understanding? There will be 92 chromosomes. Now students, anaphase is the best stage to study shape of the chromosome. Best stage to study shape of chromosome. Shape of chromosome. That is V shape, I shape, J shape, L shape. Okay? Best stage to study. Understood? Best stage. Now, let's read what is given here. At onset of anaphase, each chromosome arranged at the metaphase splits simultaneously, and the two daughter chromatids, now referred to as daughter chromosomes. The two daughter chromatid, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. They are going towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole. That meaning is that the centromere is directed towards the pole. Centromere is directed towards the pole. Yes, understanding the point? And hence at the leading edge, with the arms of the chromosomes trailing behind. Arms of the chromosomes trailing behind. They are trailing behind. Understood? So centromere towards the pole, trailing behind are the chromosome arms. Thus the anaphase stage is characterized by the following key events. Centromere splits and chromatid separate. Very first point. So if you ask me, where does the centromere split, which stage, you are supposed to tell me, sir, it is the anaphase. Anaphase is a stage where the centromere splits and the sister chromatids are pulled towards the opposite poles. Done. Chromatids now move towards the opposite poles. Chromatids are moving towards the opposite poles. Beautiful. And with that, we are coming towards the last part of the M phase now. Last part of the M phase. But chapter is not getting over. Okay. We have lots to do still. Lots to do. Okay. Now, we'll come to telophase. After telophase, we'll do something called as DNA content. You want to do or not? You want to do, right? DNA content. That is the key importance. Then we'll do significance. Then we'll do PYQs. Okay. Very students, I'm trying to go as slow as possible because yes, you have more than 250 days. So I want you to understand in depth. You cannot do such long sessions towards the end of the examination. I cannot go so slow in the examination, right? If your examination is around 2 weeks or 1 month, I cannot go this slow at that time. Now I'm making sure that everyone understands the point, write down the notes, understand each point by point of NCERT. This you cannot do later on. Later on is the revision time. That is the revision time. Now is the concept building. You're building the concepts right now, all of you. Okay, and I want all of you to clap for yourself for sitting for, do you realize you're almost sitting for almost 1 hour 45 minutes sitting with me and learning with me, and that is not very easy. That is not very easy. For you to sit in front of the computer or phone or tablet and learn continuously for 1.4 hours is very amazing. Okay, congratulations to all of you also. And trust me, all of this will be fruitful. Whatever you're learning right now is the foundation concepts for you to solve questions later on, and I will prove it to you. Okay, a telophase. Students, again, let's understand with the help of diagram. Let's understand with the help of diagram again. Okay, that is very important. Diagram is very important. Biology without diagrams is incomplete. Biology without diagrams is an incomplete story. Okay, telophase. Understand telophase. Understand the telophase first of all. Understand telophase. Okay. Now, what do we have in the middle in telophase? Let's do this metaphasic plate. Imaginary line this is, okay. Now, what exactly happens in the telophase? Remember students, telophase is the complete opposite. Telophase is the complete opposite of prophase. That point you should remember. Telophase is complete opposite of prophase. Complete opposite. Okay. It is a telophase. Telophase is the complete opposite. Now here what happens? This is the that's all. All the chromosomes, friends, all the chromosomes cluster at the poles. Obviously, all the chromosomes are clustering towards the poles now. Yes, they're clustering towards the poles. Now, once they are clustering towards the poles and they're clustering into one giant ball, they're losing their identity. They're losing their identity. See, only when they're spread apart, identity is there. So when they're clustering again, identity is lost. You tell me, when you go to temple, if the temple is heavily crowded, can you find where is your brother or sister? No, very difficult to find. If there is temple is empty, only one person is there, you can identify. This is what happens when you go to a ground. If everyone is covered, when there is lot of people together, clustered into one point, identity is lost. Identity is lost. Identity is lost. Individuality, identity is lost. And nuclear envelope, can you see? Nuclear envelope is coming back, or nuclear membrane reappears. Nuclear membrane reappears. It reappears. Nuclear membrane reappears. Nucleolus is coming back. Golgi complex, endoplasmic reticulum, everything is coming back. Golgi complex, endoplasmic reticulum, everything is again coming back. It is reappearing. Reappearing. Okay. Now here we have 46 chromosomes. Here we have 46 chromosomes. And students, remind me, tell me, here we learned, right, in anaphase, 46 chromosomes with a single chromatid. That is very important. 46 chromosome with a one chromatid, right? One chromatid. Only one chromatid. Here we had, initially we had two chromatid, but in anaphase chromosome is splitting up. So there is only a single chromatid here. Only a single chromatid is here. Okay. Now students, with that, telophase is also over. At the beginning of the final stage of chirokinesis, that is the telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. Decondensing, losing the individuality. The individual chromosomes can no longer seen, and each set of chromatin materials tends to collect at each two poles. So we have chromatin material coming back. Chromatin material coming back now. So what is happening here? Chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope develops around the chromosomes, cluster at each pole forming the two daughter nuclei. Nucleolus, Golgi complex and ER also are reforming. Everything is coming back. Any doubt you please ask me, students? Any doubt here? This is telophase. This is going to be a telophase. Now students, now we go to cytokinesis. If there is no doubt, let's go to cytokinesis now. Let's go to cytokinesis. Okay. Let's draw and understand once again. Let's draw and understand cytokinesis. Cytokinesis again is different. Cytokinesis is super different in the case of both. Cytokinesis is different in the case of animals, and cytokinesis is going to be completely different in the case of animals and plants. Animals and plants, completely different in the case of animals and plants. Cytokinesis. In the case of animals, in the case of plants. In the case of animals and the case of plants, they are completely different. Now, what exactly happens in the case of animals? Understand. In the case of animals, what is happening in the case of animals? This is the cell. This is the cell here. Okay, this is the cell here. This is the animal cell. Okay, this is the cell. Now, what exactly happens here to the cell? Okay, what exactly happens to this cell right here? This is the one nuclei here and one nuclei here. Okay. Now, here what exactly happens is, in this regions, there is some form of actin filaments called as microfilaments. Remember microfilaments from your cytoskeleton. Microfilaments. Now, when you try to take a gum or bubble gum, or if you take a balloon only, Take a balloon and try to pinch it from the both the sides. What happens? The balloon will go inside. Now if you keep on pinching, obviously the balloon will burst. If there is a very big balloon, what will happen? A small balloon will burst off. On your face only it will blast. Face also blast, balloon also blast. Okay. Imagine, keep it away from your face. Okay. Take a balloon away from your face and try to pinch it from both the sides. Pinch it, pinch it, pinch it. At one point, both the balloons will meet from the inside. The balloon will meet from the inside. Yes. That's what is going to happen here. This structure will end up looking something like this if you keep on pinching it. Yes. Will end up something looking like this. This is called as furrow formation. This is called as furrow formation. Furrow formation. Furrow formation. And if you keep on pinching it, what will happen eventually? Eventually, the two cells will become separate. The two cells will become separate with its own nuclei. With its own nuclei. With its own nuclei. Now, the furrow formation is happening from the outside. This type of furrow formation is called as centripetal manner. Centripetal. Centripetal manner. Centripetal manner. Okay. Centripetal manner from outside. Outside to inside. We are pushing it. Done. Okay. Sir, in anaphase, 92 chromosomes with one chromatid. No, sir. But you said 46 chromosomes with one chromatid. Okay, let me tell you. After this, I'll tell you that. Rashmita, I'll tell you. I told 92 chromosomes in the entire cell. And when you take it split into half, 46 on the top, 46 on the bottom. Together, 92. Together, 92. 46 on top, 46 on bottom. With one chromatid. That's all. Understood? Now, we'll come to plants now. What exactly happens in the case of plants? Now, students, in the case of animal cell, you are able to pinch it very easily because there is no cell wall. But in the case of animal cell, in the case of plant cell, above the plasma membrane, there's one more layer, right? One more layer. That is the cell wall layer. Cell wall layer is there. If the cell wall layer is present, can you pinch it now? Try pinching it from side. Nothing will happen. It was nice one gunda is sitting there. How much ever you pinch it, it's like, no, I will not bulge. How much ever pinch it, it will not bulge from the outside. So, if it is not bulging from outside, what is the other option? Tell me. If you are not able to convince your friend, if you are not able to pull your friend, what will you do? Emotional blackmail from the inside. If you are not able to pull your friend to a party, or if you are not able to pull your friend to somewhere here, what will you do? You will blackmail your friend from the inside. Emotional blackmail, right? Same your mother also does. Mother, mother will hit you one time. She'll tell you, go do your work. If you don't listen, what will your mother do? Your mother will emotional blackmail you from the inside. That's what we'll do now. If this is the nucleus, this is the nucleus here. This is the nucleus here, right? There are two nucleus after division. Now, we will do something called as cell plate formation from the inside. You realize, from the inside, we do the cell plate formation. From the inside now. Okay. Inside, we do the cell plate formation. There is something called as cell plate formation. Cell plate formation. Cell plate formation is happening. Cell plate formation is happening from the inside. Now, plate formation happens now. What will happen? Can it easily divide now, students? Can you imagine? Try to imagine. All of you close your eyes and imagine once. Okay. From here, the two cells will easily divide. Easily divide. Okay. That is called as centrifugal manner. It's called as centrifugal manner. Understood? This is centripetal from outside. This is centrifugal in the case of plant. Friends will always do, trust me, friends will always find some emotional blackmail, one ultimate thing they'll do, and you will agree. Okay, always it happens, always it happens. Let's read what is given in NCERT now. Okay, let's read what's given. Mitosis accomplished not only the segregation of duplicated chromosomes into daughter nuclei, that is chirokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm, called as cytokinesis, at the end of which the cell division gets completed. In an animal cell, this is achieved by appearance of furrow. I told you, furrow formation. Furrow formation is happening in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Diagram, students, diagram. Furrow formation, and it is joining towards the center and dividing into two cells. Okay. Now, the plant cell, however, are enclosed by a relatively inextensible cell wall. See, plant cell is like, no, I don't care. I am going to stay. I will not, you know, if you pull me also, I will not come. If you pull me also, I will not come. Only thing you should do is emotional blackmail now. I am coming here. See. Therefore, they undergo cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grow outwards to meet the exiting lateral walls. See, from the inside, it is coming towards the outside. Cell formation. Done. The formation of new cell begins with the formation of simple precursor called as cell plate. That represents the middle lamella also. Cell plate represents the middle lamella here, between the walls of the two adjacent cells. At the same time, cytoplasmic division, organelles like mitochondria and plastids gets distributed between the two daughter cells. In some organisms, very important, important line of NCERT. In some organisms, chirokinesis is not followed by cytokinesis. Imagine the cell is dividing, the nucleus is dividing, but there is no division of cytoplasm only. Imagine the nucleus is dividing, 2, 2, 4. This is one cell. In the one cell, what is happening, students? In this one cell, nucleus is dividing, but the cytoplasm is only not dividing. What will happen then? Trouble will happen. Coconut will happen. Coconut endosperm will happen. Free nuclear endosperm. Okay? Coconut will happen. See here. As a result of which, multi-nucleated, see, 1, 2, 3, 4, 5, 6 nucleus in one cell. Multi-nucleated condition arises, leading to the formation of syncytium. Okay? Syncytium. Liquid endosperm in coconut is the example. Liquid part, you know, you drink, no, liquid. Coconut, all of you like coconut water, no. That is not burst, it will not burst. It will not burst. That will form syncytium. Okay? With that, your cytokinesis is also over. Now, students, before we go to the PYQs, can we do some DNA content now? Students, it doesn't matter if the class goes long, the only thing which matters is if you are learning, that's all. The only reason I am able to stand here and teach for 2 hours is because I am enjoying this. Honestly, when you enjoy something, you will never feel tired. You will never feel like, why am I doing this? I enjoy teaching, I love it, and I don't feel tired at all. I can do this for 6 more hours, 7, 8 more hours with a smile and stand here and teach all of you. Okay. It's only a matter of if you are not willing to sit with me and learn in that process. Okay. That is the whole thing. I enjoy this because I love teaching and I'm doing what I love. And if you want to do what you love, that is going to become a doctor. This is a small sacrifice which you have to give. A small sacrifice. Someday you will also enjoy the 10-hour shift. There is something called as 48-hour shift, 52-hour shift. It will not be a burden to you because you will enjoy it, the profession which you take. It's all the matter of enjoyment. Right? So chill, it is very chill for me. Okay. Now we will do the DNA content. Okay. Now, very simple DNA content, very simple. Now tell me, in G1 phase what happened? In G1 phase, this was the chromosome. Right? This was what? 2N, 2C, 2,1cc. In S phase, what happened? The chromosome content doubled. the chromosome content became double. It became 2n4c. All of you remember? Chromosome number is same. DNA content is increasing. DNA content is increasing. Right. Now, what happened in G2? G2, nothing happened. It is going to be same. 2n4c. Now, after G2, what is there? We have the prophase. We have the prophase. In prophase also, there is no change. Prophase also, there is no change. 2n4c. 2n4c. Prophase. Now, what about metaphase? Metaphase. In metaphase also, there is no change because all the chromosomes are there as such only. 2n4c. Now comes the anaphase. Now comes the anaphase. Now see, let's go back. Let's go back to anaphase. Where is the stupid anaphase? See students, I told you, in the case of anaphase, temporarily, I told you, in the case of anaphase, temporarily there are 92 chromosomes. So n is 23, 2n is 46, 3n is 69, 4n is 92. 4n is 92. 92, 4n. So technically, can I say, students, technically can I say here we have entire thing is 4n 4c condition. DNA content will remain the same. 4c will remain same, but n is temporarily 4n here instead of 2n because of 92 chromosomes, right? 92 chromosomes. Okay. So in case of anaphase, we have it's again becoming 1. Okay. I'm talking about the entire cell. They will be separating it out now. This will become something like this because splitting of centromere will become 4n 4c temporarily. 4n 4c. Okay. 4n 4c. Anaphase. Now what happens in telophase then? Telophase. What exactly happens? In telophase there are two ways to look at it. One is per nuclei. One is per the entire cell. Understand difference. Understand nuclei and cell. You understand difference. When you look at the telophase, students, if you look at the entire cell, if you look at the entire cell, it is going to be 4n 4c because 46 chromosomes, 4n 4c. But if you look at per nuclei, because nucleus formation is happening here, chromosomes getting clustered. So if you look at the 46 chromosome, if you look at the 4n 4c, it is divided into 4n 4c is divided into 2n 2c, 2n 2c. Basically, 2 is going here, 2 is going here. Out of this 4, 2 is going here and 2 is going here. So 2 and 2c and 2 and 2c. Together 4 and 4c. Individually 2 and 2c. Individually 2 and 2c. So per nuclei it is going to be for the entire cell, we'll write first. Entire cell is going to be 4 and 4c. Entire cell. But per nuclei it is going to be 2 and 2c. Now students, understand this point now. Understand. We started off with 2 and 2c. We are ending with 2 and 2c. Can I tell it is something called as equational division then? Can I call this equational division? Why? Because the chromosome number and DNA content in the end of it, the cycle is remain the same. That's why it's called as equational division. The entire mitosis is called as equational division. Done. Equational division. Boom. That's the beauty of the science. Done. Okay. Equational division. Done. Understood? Now students, In the last 2 hours, we'll write significance also. Let's finish significance first. Mitosis or equational division is usually restricted to the diploid cells. However, in some lower plants, I told you, I think this is done, right? Some social insects haploid cells, that is drone. It is very essential to understand the significance of division of life of an organism. Are you aware? What is the significance? Okay. Mitosis usually results in the production of diploid daughter cells, right? 2N to 2 daughter cells. Both are 2N, 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 2C, right? Daughter cells with identical genetic composition. The growth of multicellular organisms is due to mitosis. Point number 1. Point number 1. Point number 1. Okay, point number 1. The cell growth results in distributing the ratio between the nucleus and cytoplasm. Point number 2. That is, when the cell is growing, because of mitosis, the cytoplasm and the chromosome number is remain the same. That is, the ploidy and cytoplasm is remain the same. 2N2C to 2N2C. 2N2C to 2N2C. Okay. That is called as nucleocytoplasmic ratio. There is because of mitosis, this nucleus cytoplasm ratio is maintained. Chirokinesis, cytokinesis. Okay. A very significant contribution of mitosis is cell repair. Third point. The cells of the upper layer of the epidermis, the cells of the lining of the gut, the blood cells are being constantly replaced. Point number 4. Replacement, repair and replacement. Repair and replacement. Mitotic division in the meristema tissue, the apical and the lateral cambium. Fifth point. The meristema tissue which keeps on dividing, the apical meristem, the lateral meristem which keeps on dividing, it is because of mitosis. It is because of mitosis. Continuous growth because of the, throughout the life because of mitosis. Understanding? All the 5 points here. All the 5 points here. Now students, we will take a 15 minutes break now. Okay. Now, after the break, let's take 20 minutes break. Only 20 minutes break. Aram se will come back. Let's take a 20 minutes break. I will also drink some water. I'll sit for some time. My legs are like, cracking little. We'll take a 20 minutes break. So all of you will refresh completely now. Full refreshment. All of you have to come. Okay. Let's take a 20 minutes break. What's the time now? The time right now is, correct, no? 8:11. Okay. 8:11 to 21, 31 will come back. 8:31 will come back again. 20 minutes break will take. All of you eat something and come in the gap. Eat something and come. Drink something and come. Okay. 8:31 will start again. Restart at 8:32. Let's take it. 8:32. Okay. Restart at 8:32. Go keep your phone for charging also. Ravindran, even I want to sit for some time. No, 2 hours I'm standing. I have to sit for some time. Okay. I'll also keep some alarm. Okay. So I'll also sit for some time, relax my legs a little. Okay. After that, we will do. There are so many PYQs. Now let me tell you the importance of it. Okay. Now you have learnt a concept, the entire cell cycle and mitosis. Don't you want to know whatever you learnt is worth it or not? Tell me. Whatever you have learnt is worth it or not? Obviously you want to know, right? It's worth it or not. So for that very reason, we'll take a 20 minutes break and we'll come back. Okay? To know that if you have whatever you have learnt is worth it or not till now. Promise me all of you will come back. Okay? So with that, we'll come back. Okay? 20 minutes break. Hello. Welcome back. Welcome back to the amazing class of cell cycle and cell division. One thing, let me refresh. Let me check if your attendance is marked or not. Yes, still not marked, huh? Nice. This only you do. Okay. Let's start now, students. Let's start. Can we start now? Yes. Let's start. First question on your board, try answering the question. First question, let's start. Namaskara, welcome back. Yes, first question is here. The spindle fiber attached to kinetochore of a chromosome during which phase? When do they attach? That is, if this is a centromere, this is the sister chromatids. Here we have a proteinaceous layer. All the spindle fibers are attached in the kinetochore. At which phase? What is the answer? Quickly. Yes, exactly. It's going to be your metaphase. At a metaphase, all the spindle fibers are attached to the kinetochore and at the equatorial plate. Right. Next question. Following are the stages of cell cycle. You're supposed to tell me which is the first, second, third, fourth stage. Okay. Now, obviously, gap 1 will be the first. Then we have gap 2. Then we have synthesis phase, which is the S phase. After that, we have the chirokinesis. Last, we have the cytokinesis. Okay. So, what is the order? What is the order then? What is going to be the order? That is going to be A. Oh, sorry. One second. What did I do? 1, 2, 3, 4, 5. Now it's proper. So E C E. Okay, wait. E C A D B. Third one. Yes. Next question. What exactly happens in all the following? Match the following. Simple question. Time starts now. Go. Please start quickly. Time starts now. Yeah, I didn't see. I was just marking it randomly. G1. What exactly happens in G1? Okay, very easy. What happens in S? What happens in G2? What happens in your G0? Time starts now. Go quickly. You have 10 more seconds to answer this question. 10 more seconds. Friends, it's very easy. Always go for what. See, every single match the following. Always go for what you know. All of you know the replication of DNA happens in the S phase, right? So B is going to be 2. So can I eliminate these two? If B is going to be 2, can I eliminate these 2? Yes, I can eliminate. You also know that the cell is metabolically active but no proliferation. That is going to be a G0, right? So G0 is going to be 1, right? C is going to be 1, B is going to be 2. There is only one option there. Right? If your C is going to be 1 and S is going to be 2, there is only one option left. Option number 2. Do you realize? Right? If your S is going to be 2 and your G0 is going to be 1, that is metabolically active but no proliferation required. So that is the cell is going to be metabolically active but it is inactive at the same time. Inactive cell but metabolically active. Right? So C is going to be 1, obviously. Then what do we have? A is going to be 4. Cell is metabolically active but grows continuously. And D is going to be 3. Synthesis of protein. How many of you got it wrong? Tell me honestly. How many of you got this question wrong? Quickly raise your hands. All of you quickly raise your hands. How many of you got this question wrong? Always go for what you know. 100% in match the following. Okay. Next question. Among eukaryotes, replication of DNA takes place in 10 seconds. Questions go. 10 seconds. You have just 10 seconds to answer this question. Just 10 seconds and the time is up. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Replication happens in your S phase. We learned it couple of times. Replication of DNA happens in the S phase. Easy question. That is DNA replication or DNA duplication. You can tell DNA duplication also. DNA duplication. DNA duplication also you can say. Okay, S phase. Option number 3. Next question. Again, students, match the following. Always go for which you know the best. Which you know the best. Okay. Now, what do you know in this following? Which one do you think is the most appropriate one? Inactive phase. Inactive phase is going to be a G0 quiescent stage. You know it very well. You know it very well. Okay. So C is going to be 2. C is going to be 2. That you know very well. Okay. Then you know equational division. That is basically your M phase is called as equational division. Yes. M phase is called as equational division. Now, we know a protein synthesis happens in the case of your G2. G2 is going to be one protein synthesis mostly. Just now learned. And we left with G1. G1 is interval between mitosis and initiation DNA replication. That is, we have M phase, we have interphase, again we have M phase. Here we have your G1, we have S, and we have G2. We have G2. So G1 is the stage. G1 is the phase which is between your mitosis and initiation of DNA replication. Initiation of DNA replication. Easy answer. Next question. Students, do you realize many questions will be from your statement based as well as your match the following. Statement based question. During G0 phase of cell cycle, the cell is metabolically inactive, students. Is it inactive or active? Tell me in the chat. Inactive or active? The centrosome undergoes duplication during the S phase of the interphase. Yes, in the cytoplasm, S phase centrosome duplicate. Statement 1 is incorrect, statement 2 is correct. Done. Done. It is metabolically active. The cell is going to be metabolically, the G0 in the active. The cell may be inactive, but metabolically it will be active. Understood? Metabolically it will be active. So statement number 1. Next question. Which of the following is called as quiescent stage of cell cycle? Again, G0. This is a very bacha question. G0 phase. G0. G0. Okay. Next question. Students, are you able to solve the questions now? Are you able to solve the questions? See, sometimes you might think the question is a little difficult because the concept you know now. It is only of how you are applying it. In G0 phase, it is metabolically active. The cell may be inactive, but metabolically it will be active only. Okay. Next question. Which of the following never occurs during mitotic cell division? Never occurs. Okay. Coiling and condensation of chromatid occurs. Spindle fibers attached to kinetochore, metaphase, yes. Movement of centrioles towards the opposite poles, yes, in the prophase only. Pairing of homologous chromosomes, it does not happen. It does not happen. Pairing of homologous chromosomes happens in the case of your meiosis. So this is never happens. This never happens. This never happens. Next question. Select the incorrect statement with reference to mitosis. Incorrect statement again. Incorrect statement regards to mitosis. Splitting of centromere occurs at anaphase, yes. At all the chromosomes lie in the equator at the metaphase, yes. Spindle fibers attached to centromere. Spindle fibers attached to the centromere of the chromosome? No. They attach to the kinetochore. They don't attach to the centromere. They attach to the kinetochore, not centromere, right? Chromosomes decondense at telophase. Yes, they completely decondense. So does not happen as incorrect as option number 3, because it is supposed to be kinetochore, not centromere. It's supposed to be kinetochore, not centromere. Okay. Next question. With respect to metaphase, which of the following statement is again incorrect? Incorrect statement. So students, they will ask you a lot of statement based questions. Match the following. Remember, match the following statement based question. Tanuja, Abhi, read the question carefully once. Okay. Incorrect statement. Complete disintegration of nuclear envelope takes place, yes. Chromosomes are highly condensed, yes, in metaphase. Okay. Metaphase chromosomes are made up of 4 sister chromatids held together by centromere. No. Why no? Because it is 4 here. It's supposed to be 2. Chromosomes lie at the equator of the cell. Yes. So what is the answer? Option number 3. Correct. Next question. Take your time and read the question once. I think I'm not giving you time. I'll move aside, read the question once. I'll move aside, read the question. The centrioles undergo duplication when? Again, very easy question, students. Very easy question. The centrioles duplicate under when? All of you know. The centrioles duplicate in the S phase along with DNA replication. So what is the before question, sir? Before question here. The question says that with respect to metaphase, which of the following statement is incorrect? Obviously, you know, right? Will there be 4 sister chromatids? Do you find some special? This is 2 sister chromatids, right? 2 sister chromatids. You will find this in metaphase. You will never find. Do you find something like this? 4 sister chromatids? No, you will never find that. Okay, understood? This is S. Next question. During which phase of mitosis do the chromatids become chromosomes? Chromatids become chromosome. Easy question again. It's only a matter of how you approach it. Absolutely correct. Did all of you understand this question? Did all of you understand this question? Did all of you understand this question? When I was teaching you about anaphase, I told you chromatid will act like chromosome. Did I mention or not? All of you tell me. Did I tell you in anaphase chromatid will act like chromosome? Daughter chromosomes. Yes, daughter chromosomes. Here, anaphase, daughter chromosomes. This was a very, if you know the proper concept, you'll be able to answer these questions. Concept level question this is. Okay. Next question, AR question. Some cells enter G0 phase leading to the inactivation of the cell cycle. Right? Inactivation cell cycle. Reason, G0 phase occurs due to non-availability of mitogen. Now understand what is the meaning of mitogen. Mitogen is basically a some type of hormone or chemical which induces the cell cycle. If mitogen is absent, for example, concanavalin A, you learn, right, secondary metabolite. Concanavalin A is a type of plant mitogen which induces the cell cycle. If mitogen is absent, the cell will undergo G0. Understood? If the mitogen is absent, cell will undergo G0 also. Non-availability of mitogen, energy-rich compound. Yes, and energy compound. Obviously, both are true. A and true. That's why it undergoes. Understood? A. Understood, right? Good question. This is a good question. Oh, that's all. Huh? How many questions did we do? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 questions. So fast. So fast, 14 questions over. Oh, so fast. So fast, 14 to 15 questions are over. Even I wanted more. I thought there were more questions. Students, I think I downloaded the wrong PDF, I think. But never mind. I will ask you some questions on my own. I'll ask you questions on my own. Okay. Tell me in which phase of cell cycle reappearing of a nuclear envelope takes place. Which phase of cell cycle reappearing of nuclear member takes place? Tell me. Quickly. Reappearing of nuclear envelope. Quickly. So fast, I'm surprised. I think you're solving very fast now. Telophase, exactly. Now next question, rapid fire, quickly answer. I want quick answers. Okay. In which stage morphology of chromosomes can be studied very well? In which phase morphology of chromosomes can be studied really well? Morphology of chromosomes can be studied really well. Correct, exactly. It is going to be your Metaphase, because complete condensation takes place. Complete condensation of chromosomes happens in metaphase. That's why complete morphology can be studied. Okay. In which stage of cell cycle shape can be studied very easily? Tell me. Shape of the chromosomes can be studied very easily, not telophase. Shape can be studied very well. That V shape, J shape, I shape, right? Where can be studied? Exactly, anaphase. Exactly, anaphase, shape of the chromosomes. Anaphase. Beautiful. Nice. Next question. Next question. The centrioles also very easy. Students, concept is very easy. All of you know the concept now. All of you know the concept. You can tell very easily now. In which phase of cell cycle? In which phase of cell cycle in which phase of cell cycle mitotic apparatus formation takes place? Tell me. Mitotic apparatus formation. Which phase of cell cycle mitotic apparatus takes place? Formation of mitotic apparatus. Formation of mitotic apparatus. And mitotic apparatus is made up of what? Mitotic apparatus is made up of what? Two things. Correct. Mitotic apparatus is made up of your asters as well as spindle fibers. Mitotic early prophase made up of mitotic apparatus is made up of asters as well as spindle fibers. Prophase, early prophase. Nice, not metaphase. Not metaphase. Prophase. Prophase. Early prophase. Nice. Next question. Next question. Let me think. I have to make questions on spot now. What is the time duration of a cell cycle in the case of yeast? Go. In the case of yeast. In the case of yeast. Tell me. In the case of yeast. 90 minutes. Exactly. 90 minutes. Okay. Next question. Next question. Repair and reform, they are significance of mitosis. True or false? True or false? Quickly. True or false? Repair and formation of new organelles. True or false? True. Exactly true. In the 24 hours of cell cycle of human cell, how much of time is taken for the interphase? Next question. Next question. How much of time is taken for the interphase? Out of the 24 hours in the cell cycle of human cell, how much of time is taken for interphase? Exactly. It's going to be 23 hours or 95 percentage. 23 hours or 95 percentage. Nice. 23 hours or 95 percentage. Nice, nice. Next question. Next question. Cell plate formation takes place in the case of animal or plant cell. Cell plate formation takes place in the case of animal or plant cell. One more point I forgot to tell you, the cell plate formation takes place because of phragmoplast. Not in your syllabus, I didn't teach you, that's why. Phragmoplast, it is because of Golgi complex. Okay. Tell the answer quickly. Plant cell, exactly. Now, where do you find this equatorial plate? What is the other name for equatorial plate? What is the other name for equatorial plate? Other name for equatorial plate. Other name. And this plant cell, you told me plate formation, right? Plate formation is a centrifugal or centripetal? The plate formation is centripetal or centrifugal? I am bombarded. I can keep walking, students. I can keep walking. I can keep making questions. That is one of my talents. I can keep walking and I can keep on making questions for you. Okay. Now, if a cell is haploid, how many kinetochores will be present? If a cell, centrifugal, centripetal, tell me. Centrifugal or centripetal? Anusha? Huh? Centripetal is from outside, centrifugal is from the inside. Cell plate fugal, inside it happens, not centripetal. Centripetal is from outside. If a cell is haploid, how many kinetochores will be there? Tell me. If a cell is haploid, how many kinetochores will be present? Centrifugal. No, Anusha, centrifugal. 46. Exactly. 23 chromosomes. So 46, 23 chromosomes. It's haploid cell, 23 chromosomes. Oh, Ramdani, 26 chromosomes are there. So double will be the kinetochore. Okay. I can ask you one more question. In a diploid cell, kinetochore, oh, you understood, it's easy. Next question I'll ask you. In the case of metaphase, spindle fibers are attaching to how many kinetochores? In the case of metaphase, spindle fibers are attached to how many kinetochores? 1 or 2? 2. This will change in the case of meiosis, students. In the case of meiosis, one spindle fiber will attach to only one kinetochore. One spindle fiber will attach to only one kinetochore. Okay? That will change in meiosis. I think we'll stop it, students. If you want to, students, I'll tell you honestly. In this NEET journey, right, I'm telling you, honestly, I'm telling you, the best thing is going to be solving questions for you. Best thing is for solving questions. And if you want to solve more questions, I would recommend you to join a test series. That is very important. You might be like, no sir, no test series, I will do out on my own. No. It is very important that you join a test series which is year-long test series. Very important. Anywhere. Okay. And if you want to join a test series from Vedantu, in the description there is a link. You can go join there. Okay. Test series is very important. The more questions you solve, more confidence you will become, and you need to solve long questions, not one short. When I say long, I mean full test, full syllabus, physics, chemistry, bio. It's very important, and you will not realize it now. At the end, you will realize it. And the earlier you start question solving, no, it's more beneficial for you. So if you want test series from Vedantu side, okay, description if you go, you will find one. That's all I'm going to say. Okay. So with that, today's class is going to get over, and thank you so much, students, for all your patience. And today's class was really fun for me. So shower your love in the comment section. That's all I can say. So all the students who are watching it recorded, smash the like button first of all, and let me know in the comment section about today's class, because part 2 is coming. And are you excited for part 2? Part 2 will have your meiosis completely, meiosis. And if you think mitosis is easy, meiosis is even more easy. If mitosis is easy, meiosis is going to be even more easy. Don't take tension. I am here to make it easy for you. DNA content also I'll teach you there. I'll make you solve questions also. Every single point I'll make you cover. So have trust in us. That's all I say. Have trust in Vedantu. Okay. So take care, students. I'll see you in the next class. Let me know in the comment section. Do not forget. If you forget, I'll catch hold of you. Pavitra, Soni, Ravindrani, Paul, Rishmita, Soni, Bandravini, Jensli, every single one. I want to see everyone's comments today. Okay. So with that being said, take care all of you. Bye and have